Now, Mendel was working with a much smaller number of chromosomes, and so he was only actually looking at one trait or so at a time. The way that we can look at this mathematically was done by a mathematician named Punnett, and he used, to represent the dominant trait, he used a character letter, capital letter, and for the recessive trait, he used a lowercase letter. So for tall, tall being dominant is short, we use a T for tall, capital T, and then we don't use an S for short because they're both the same gene. So they both need the same letter, but we can use a lowercase letter to represent the recessive version of that gene. Okay? So the capital letter is dominant, lowercase is recessive. Same letter, depending on the gene. And now, every cell has two copies of each gene, right? It might have two capital T's, a capital T and a lowercase t, or two lowercase t's. But when that cell forms gametes, if it's a germ cell and it forms gametes, those letters are separated from each other. And so each gamete gets one letter, either a capital T or lowercase t, depending on what the parent plant looks like. Okay? Then we take a gamete from two different plants, each with its one letter, and we put them together to form a new organism with two letters again. Okay? Remember, you need two copies of each chromosome to live. So we put that together again, and then depending on the specific combination of letters, we'll figure out what the offspring will look like. So the term haploid we use to talk about gametes that have half the number of chromosomes, one copy of each. And diploid means gamete, or a cell that has two copies of each. So gametes are haploid. They contain one copy of each chromosome. When they meet at fertilization, two gametes meet at fertilization to create a diploid zygote, a new organism. And it will have its two copies of the gene, and those two copies will determine what it looks like. Okay? There are three possible possibilities here. We've got two capital letters, capital T, capital T. If we're talking T means tall, this is two dominant tall genes. It's going to be a tall plant. We call that homozygous dominant, homo meaning the same, right? Homozygous dominant. We could have two lowercase. Okay? Remember, lowercase t represents short. If you've got two of these, there's no dominant gene, there's no tall gene. This is going to be a short plant, homozygous recessive. But what if it gets one of each? If it gets one of each, capital T from one parent and a lowercase t from the other parent, we call it heterozygous. Hetero means different, or hybrid. What is it going to be, tall or short? Well, because it's got a dominant gene in there, one of the copies is dominant, that dominant gene will dominate over the short gene, and so it'll be a tall plant. Okay? The phenotype, what it looks like, doesn't necessarily tell you what its genes are, except for the homozygous recessive. If you have a short plant, you know that it's got two copies of the short gene. That's the only one you know for sure. But if you have a tall plant, you don't know if both copies are dominant or if only one copy is dominant. Okay? You just can't tell. So if we use these letter designations, we can follow how a trait might be transmitted. And really what we're looking for is what are the frequencies for the offspring. Okay? So remember, gametes, when they meet at fertilization, it's a random combination. Okay? And it's only the combination that of those two letters that determines what the offspring is going to look like. So if a T, capital T sperm meets a capital T oocyte egg, you'll get homozygous dominant, and so on and so forth. Okay? And Punnett developed a mathematical way of looking at the possible combinations. It's called a Punnett square. Okay? This is what it looks like. This is a monohybrid cross. So what Mendel did was he took homozygous dominant plant, capital T, that's true breeding. When that plant crosses with anything, it will always produce a tall plant because it's always going to give one of its capital T's. It's tall genes. And a true breeding homozygous recessive plant, lowercase t, that plant, if you cross it with other short plants, will always give short plants. It's true breeding for short. All four possible combinations, we've separated out the two genes at gamete formation. We've separated them out, and so each time you fertilize, you get one copy. The top parent here always gives a capital T, and the one on the left always gives a lowercase t, so the offspring will always have one of each, which means they'll all be tall, but they'll all be heterozygous. They'll all be hybrids. Okay? Now, if we take two of those and we cross them with each other, we get the second generation. And now things get interesting because each of those hybrids will either give a capital or a lowercase t, a tall gene or a short gene. And depending on how it combines with the other plant's genes, we might get tall or short plants. So you can see here we get two capitals together. Each of the two parent plants gives a capital. We get a tall homozygous dominant plant. If one parent gives a lowercase and the other one gives a capital, one gives the short gene, one gives the tall gene, you'll still have tall plants, but they'll be hybrids. And finally, if both parents give the short gene, the lowercase t, then you'll get a short plant. And if you look at the ratio, it's 3 times as many possible tall plants as short plants. That's the 3-to-1 ratio, the phenotypic ratio that Mendel found. Okay? Now, sometimes Mendel didn't know what a particular plant was. Okay? When do you think that would happen? Well, if it was homozygous or heterozygous. If it's heterozygous, you don't know by looking at it. It looks tall, but you don't know if it's got 2 tall genes or a tall and a short gene. You don't know. He could do a test cross. And the way you do a test cross is you cross the unknown with the homozygous recessive version of that plant. And depending on how the offspring looked, you could tell what the original parent was. Okay? So it looks like this. The left side is the homozygous recessive. That's the one we know for sure. We know it's homozygous recessive because it's going to be a short plant. But we don't know if the top is capital T on the left or capital T lowercase t on the right. Depending on what the offspring look like, we can tell. If we get only tall offspring, then that means that the unknown plant, the unknown tall plant, must have been homozygous dominant because there's no way that you could produce a short plant if that's the parent. But if we do get short plants, and it's like 50-50 over here on the right, then that means that the unknown tall plant at the top was heterozygous because it was able to produce short plants with another short plant. That's what a test cross does. It tells you what the particular genotype of an unknown plant was, especially if it has the dominant trait.